Hello, our project is titled Nutrient Management, Heterotrophic Nitrification. Our team members are Zach McClellan, Jaden Storer, Anna Scribner, and myself, Jace Bradley. We'd like to thank our industry sponsor, West Tech, as well as Utah State University. Nitrogen removal is very important in wastewater treatment, as too much nitrogen can lead to harmful algal blooms and other public health concerns. The current industry standard is removing nitrogen in two separate steps, nitrification and denitrification. These two steps are done by two different organisms and can be quite costly. Our project is focused on doing those two steps with one organism named Alcaligenes faecalis. A previous senior design group was able to determine mathematical constants for Alcaligenes faecalis, and that would allow us to relate its nitrogen removal rate to substrate concentration. This was done in a synthetic wastewater, and our project is focused on expanding that research.
Preliminarily, we wanted to determine if A. faecalis can remove ammonia at a rate sufficient enough that it could even feasibly be used in wastewater treatment. We designed an experiment where over a time span of 6 hours, we looked at ammonia removal of A. faecalis in synthetic wastewater and while it is still in the exponential phase. We decided to test with 3 different methods with A. faecalis in a rope media, a moving bed bioreactor or MBBR media, and just a standard suspension media. In figure 1, we see results of this experiment, and we see 3 things. 1, that there wasn't too much a difference between the different methods. 2, that it was able to remove ammonia. It went from over the time span of 6 hours all the way down to under 2 milligrams per liter. And 3, that it did so in a very linear fashion.
Next, one of our focuses was to see if A. faecalis could grow biofilm in either synthetic or real wastewater environments. We tested this by using the MBBR media and growing it for a period of 3 weeks to try to grow this biofilm. We then transferred the media into another flask and tested the ammonia removal in the second flask. The results are shown here in figure 2, which show that only one of the samples, which is the wastewater growth, showed a significant ammonia removal within the first 9 hours. This suggests that there is some kind of biofilm that retains A. faecalis as it was transferred. Figure 3 shows the microscopy scrapings of these MBBR media, again supporting that there was more bacterial growth on the real world or the real wastewater media.
To summarize the results that we obtained, we found that when A. faecalis was grown in exponential phase in a synthetic wastewater environment, it removes ammonia at a 0-order reaction rate, and it can remove ammonia from a concentration of 40 milligrams per liter to below 2 milligrams per liter in under 6 hours. We also found that there wasn't a significant difference between the different growth methods that were used. We also found, as Zach explained, that A. faecalis can potentially grow a symbiotic biofilm with an unknown organism in real wastewater. This leads into future work that can be done in this project, in which we would like to find and isolate the organism that A. faecalis can form a biofilm with, and we would also like to further test the capabilities of A. faecalis to grow a biofilm on its own. We would also like to test the effect of other conditions on the growth and ammonia removal of A. faecalis, such as the effect of temperature. Finally, we would like to test the kinetic constants as a previous group found, but instead of finding them in a synthetic environment as they did, we would like to redo that experiment in a real wastewater environment. Thank you.